Hello everyone, I'm Dr. Karami from Iran. I'm a biology teacher at Abhishek Coaching. Today we'll discuss the topic cell structure and function for grade 9. Let's go. Okay, the most important function of the cell is making proteins. And we have to know the process of making proteins to understand what is the function of each and every organelle in the cell. So that's why I want to explain why and how cell makes proteins. Cells use genes to make proteins for critical jobs like carrying oxygen and contracting muscles. Explore the steps of protein synthesis, the process by which DNA is transcribed and translated into tens and thousands of different types of proteins that make us what we are. So, as you understood from this text, the process of making proteins, it happens in the nucleus of the cell, and it is done by DNA, which is the genetic material inside of the nucleus. In this picture, you can see nucleus in the cell, and here is the double-stranded DNA, which from this, the process of transcription occurs. And here you can see the process of transcription, and after that we have mRNA to make proteins. Okay, mRNA is needed to make proteins. Here, as you see, is nucleus, and here is cytoplasm. So after this mRNA is translated by the organelle, which is called ribosome. So ribosome has the function of protein synthesis. Okay. As you see, here is the ribosome. Okay, here is the ribosome, and here we can see the protein which is made by this process. Okay, here I won't explain the process of transcription and translation because it's very long and it's not for now. Let's go to the next. So types of protein made in the cell. There are a total of 7 different protein types. These include antibodies, contractile proteins, enzymes, hormonal proteins, structural proteins, storage proteins, and transport proteins. So there are many different proteins in our body, like different hormones, different enzymes, even hemoglobin, which you can see in this picture. So these are different proteins which are made by cell. That's why it's very important to understand that cells, at first, they make proteins, and then how, by the help of which organelles they make these proteins. This is important to understand, okay? If you understand the process of making proteins, and if you understand which organelles are doing this, so we can easily remember during exam, we can easily remember that, okay, which organelle has which function, right? So let's go to the next. Ribosomes. When a cell needs to make proteins, it looks for ribosomes. Ribosomes are the protein builders or the protein synthesizers of the cell. So please pay attention that ribosomes are protein synthesizers of the cell. They are like construction guy who connect one amino acid at a time and build long chains. Ribosomes are special because they are found in both prokaryotes and eukaryotes. Okay, remember this is very important. Okay, in this slide you can see the one cell. You can see structure of one cell, and what is cytoplasm? Cytoplasm is a jelly-like fluid which is inside of the cell, and all organelles are floating around in this jelly-like fluid. Okay, so we have inside of the cell, except cytoplasm, and inside of cytoplasm we have different organelles like endoplasmic reticulum, which we write it as ER, short form as ER. We have Golgi apparatus, we have nucleolus, mitochondria, lysosomes, and secretory granules maybe, and microfilaments and microtubules. Okay, so these are different organelles inside the cell. Now let's go and see what's the function of each and every of these organelles to make proteins at first. So the most important part to make protein is nucleus. In the nucleus we have a process of transcription, and so process of making proteins start from here. Also nucleolus, it makes ribosomes. So ribosomes are made in nucleolus and then leave the nucleolus and then they leave it, and some of them they freely float around in cytoplasm, and some of them they attach to ER or endoplasmic reticulum. That's why we have two types of endoplasmic reticulum. We have smooth endoplasmic reticulum without any ribosomes, like this part in this picture is this part, okay? And second type is rough endoplasmic reticulum, in which ribosomes are attached to endoplasmic reticulum, okay? Then, okay, so from the beginning, ribosomes made here, they leave the nucleus, they attach to endoplasmic reticulum. Now, what's the function of endoplasmic reticulum? Endoplasmic reticulum, as you see from the picture, you can understand what's the function. So here you could see some passageway, right? It's like some passageway. So the function is the same as what you see. Endoplasmic reticulum function is a passageway for proteins which are made by ribosomes, okay? So proteins pass away from which made by ribosomes from this part, and by the help of some vesicles, in this picture you can see, they leave the endoplasmic reticulum, and then they migrate to Golgi apparatus, right? To Golgi apparatus. Maybe I can move this my picture to the side. So in the Golgi apparatus, then now what's the function of Golgi apparatus? The most important function of Golgi apparatus is to fold proteins. So the function of Golgi apparatus is to fold and pack the proteins, because without folding and without packing, it's not possible for the cell and for the body to use those proteins, okay? So remember, Golgi apparatus, just folding, okay? And after that, protein can leave this place to do its job, okay?